So the forebrain is essentially what we usually think of when we think of our brains. And it consists of this wrinkly surface area. And we divide this into the frontal lobe, which is the largest lobe, the parietal lobe, the occipital lobe, which is at the back of the brain, and the temporal lobe at the sides. So once again, we're looking at a side view of the brain. Here's our brain stem. The cerebellum is here. All of this is referred to as the cerebral cortex, and it's divided into, as I said, these 4 different lobes or cortices. Each of these lobes has a specified function. It has a smaller subdivision in it that typically specializes in one thing. For example, the occipital lobe specializes in vision. The temporal lobe specializes in auditory sensation, so hearing, but it's also typically where we think we store long-term memories. The parietal lobe, it has a sensation called somatosensory, and that's about where you exist in space relative to other things. So that's what is failing when you stub your toe on something or you smack your leg into a desk as you try to walk by. We're going to come back to the frontal lobe because the frontal lobes, very important for some of our areas when we talk about sleep and waking, and also important in something called executive function. Now, once again, we've got that brain stem coming up into the middle. And here's the trick. Also in the middle of that area, which we can't see, we have groups of structures, a group of structures that we refer to as the limbic system. The limbic system contains a lot of different things, and some neuroanatomists put in some structures that other neuroanatomists don't. We care about it for 3 different things. We care about it for the hippocampus. Don't confuse the hippocampus with the hypothalamus. The hypothalamus is adjacent to the thalamus in the diencephalon. The hypothalamus is about hormonal control. The hippocampus is about learning and memory. I like to think of the hippocampus as the grand associator. It is what allows us to remember that you wore that dress or that pair of pants at this social event and you talked to these people and this is what they said. But it's also what allows us to remember that there's 4 things or cortices in the forebrain. So we're grouping things or associating things together. The hippocampus is a bilateral structure existing on both sides of the brain and it lies right underneath that temporal lobe. The amygdala lies adjacent to the hippocampus and it's where emotions are stored, particularly emotions related to fear and anxiety. So if you want to try to remember what the amygdala is, A for amygdala, A for anxiety. And remember, anxiety is just fear for something that isn't present. So fear is the appropriate response when there's a tiger about to get you. Anxiety is when you're afraid that there might be tigers while you walk to work in the morning. And so the fear for something that's not right in front of you. And then we have a structure called the cingulate cortex, which is another, just a bunch or a region of the brain. And it helps communicate between the limbic system and the basal forebrain, which is this area right at the bottom of the frontal lobe. So let's take some other images. So once again, we've got our mid-sagittal views over here on the right. And then this is kind of an interesting image from the posterior. So we can see the posterior. Remember, here's dorsal. And then we can't quite see anterior. But what I like about this is you can see that here's our temporal lobe. And then underneath that temporal lobe, so these things are hiding underneath it, is our amygdala. And then this hippocampus. And this happens on both sides of your brain. And in the middle is this basal forebrain that's almost adjacent or right in back of, posterior to the prefrontal cortex. And then, of course, we have this mid-sagittal view, which you've already seen in multiple contexts. And so here's our thalamus, here's our hypothalamus, there's that little pituitary stalk sticking down. The amygdala is overlaying those structures, and then the hippocampus is existing it has this shape to it that's a horseshoe almost shape to it. So mid-sagittal view, this is a nice CAT scan version, and then we see our basal forebrain, and this right here, this white structure, we've got our cingulate gyrus and cingulate cortex. So could be the hard to make out, but that's generally what we're looking at, once again, in the middle of the brain. Now don't worry, there's not going to be an exam where I put up an image of the brain and you have to figure out which structure is which. What I'm most concerned with is about you realizing that these structures are close together so they can interact easily, and also which structures do which things. Speaking of which, the frontal lobes, what do? They are in charge of executive function. And so executive function is, how do we control what we do? How do we decide what to do? Do we decide that we're going to eat that piece of chocolate cake, or we're going to stick to our diet and not eat that chocolate cake? And so we look at executive function being things like, what am I going to pay attention to in my environment? How do I decide what to do with my environment? And then what would be the effects of those things? And so problem solving, cause and effect. If I'm going to plan to do something, I also have to plan the movements involved in it. So it controls and coordinates movement. And then we have a lot about social interaction. And the frontal lobe contains that primary motor cortex at the very back. So this red zone is actually part of the frontal lobe. And that primary motor cortex is really concerned with controlling your hands, your feet, and your face, because your face is really important for social interaction, right? Are you going to smile? You got to read other people's faces. Are you going to mimic what they're doing? And then our hands, really important for fine motor control. And the frontal lobe basically also coordinates where we're spending our attention. So when we're multitasking, which we can never do too successfully, we always fail a little bit at multitasking, but basically our ability to switch back and forth between tasks, that's our frontal lobe. Okay, so we've covered a lot of brain areas, and what we really want to know is, what are the important ones for sleep and wake, and what do they do? Once again, a mid-sagittal view. This is from another textbook that I pulled out because I happen to like the color images more than what we have in our textbook. And what we've got is that ventrolateral preoptic nucleus, VLPO, in the basal forebrain area. Look at that. And that basal forebrain area is going to be really important for triggering sleep. And that sleep is going to be triggered by levels of adenosine. All right. Keeping you awake, we have things like the reticular formation, the locus coeruleus with our norepinephrine. Here we got yet another portion of the reticular formation, and the thalamus. And so we've got the Raphane nucleus probably be very important in kind of the balance between REM, waking, and non-REM sleep. So we'll come back to that. All right. Reticular formation is big. It runs the length of the brainstem. So the front part, anterior, the anterior reticular activating system, it's what maintains wakefulness and coordinates all the things that we need to pay attention to our environment. So the reticular activating system, put anterior in front of it because it's the front part, it helps coordinate glutamate, norepinephrine, which I think has an E. We just have lost it. There it is. And acetylcholine. It brings it helps integrate and bring sensory information into the thalamus. Remember, the thalamus is about all sensory information. So what's happening in our environment, what's touching our skin, what do we smell, what do we hear? It coordinates with the hypothalamus, that basal forebrain area, And then, of course, it's adjacent to, because it's in the brainstem, norepinephrine and serotonin centers with regard to the locus coeruleus and the raphe nucleus. So the thalamus is pretty cool, actually, because, yes, it's a sensory warehouse, but what it does is it also filters what sensations make it into conscious awareness. And you might say to yourself, well, shouldn't all my sensations get into conscious awareness? Well, when you first put clothes on in the morning, that's a change. Your thalamus lets you become aware of that sensation, but then hopefully you kind of forget about it and you stop paying attention to it. And so the thalamus acts as a gatekeeper, letting your upper brain or cerebral cortex be aware of, be told what it needs. And it also coordinates with some of the sensation centers that are happening up in those cortical areas, like the primary visual system, the primary auditory system, and the somatosensory. So we have information that's coming up viscerally through the body. The thalamus doesn't necessarily relay all of those upward, and then it helps coordinate with the information that's coming down from the brain. So the thalamus is all about sensation, sensory processing. It's not just a warehouse, it's a distribution center. And so it passes that sensory information up or down as needed. Up or helps integrate coming down. So during REM sleep, the thalamus keeps that cortex activated, but it prevents sensory information from the world waking us up. And in fact, what we see is as we progress through the stages of sleep, we get less and less responses to exterior sensations. And so if you wanted to sneak up on somebody during their sleep cycle, it's actually better to do it in, say, the 3 to 4 a.m. when more of their cycle is in REM sleep, because only 10% of stimuli trigger a cortical response, as opposed to 65% during N3 or some of the deeper stages of slow wave sleep. So once again, during REM sleep, that thalamus helps kind of keep the cortex activated because we have to have the set of our dreams. We have to have information that's coming that we're going to use for our dreams. And that's usually sensation or sensory information. And then the thalamus helps maintain a low, slow EEG for non-REM sleep. So once again, to turn on sleep, the basal forebrain, specifically the VLPO, is going to help coordinate with the hypothalamus and other areas the switch for sleep. It's going to basically become triggered because of adenosine. It's going to trigger in turn GABA, which is going to inhibit the anterior reticular activating system and the areas of the hypothalamus, because the hypothalamus has a lot of different sub areas. Those areas of the hypothalamus that deal with waking. Remember, we had histamine, we had auroxin because of those the narcolepsy conditions. Okay, that's all hypothalamus. And so the VLPO coordinates with all of that. So essentially we have this flip-flop. We flip a switch and we get sleepy, we're asleep and we flip back and we're awake. And having in-between states is actually bad. That's where we get some sleep disorders. Things like hypnagogic hallucinations, where people report dream images coming into their waking state. If you saw the Netflix show Haunting on Hill House, there's that whole sequence where the one character has muscle paralysis when she wakes up and she sees a dark image or person in the room with her. So me as a sleep person, I'm like, well, she may have muscle paralysis from her REM states, and then she has some hypnagogic hallucinations. So sometimes I'm not fun to watch horror movies with. But what I'm suggesting is that what happens is a sleep disorder when we don't have a clear flip-flop circuit, when these elements of sleep intrude into our waking state. And in fact, how do we know that the basal forebrain is really important for putting us to sleep? Well, lesions of it actually produce insomnia, and a lesion is a damage. So if we damage that basal forebrain, people report insomnia. And in fact, in Alzheimer's, we see damage to that area that's correlated with the sleep difficulties that individuals with Alzheimer's have. Okay, so the VLPO actively inhibits all these arousal systems. But here's the trick. For REM, we've got to turn some things back on to dream. And so the VLPO is going to monitor sleep debt. It's going to promote sleep. It's going to, with the thalamus, it's going to basically coordinate how sensory information is processed and how we can maintain kind of a slow, steady sleep state that keeps things in the outside world from intruding. And then we're going to shift into REM. And so what's going to happen is we're actually going to have two groups of cells. We're going to have what we call REM off cells and REM on cells. And they appear to be clusters of cells that behave differently in the brainstem. Okay? So REM off cells, those occur in the raphe nucleus, and they help maintain non-REM sleep. So in other words, they are saying you need sleep. You don't need to dream right now. And so serotonin and histamine, they're both active during waking and non-REM, and they inhibit REM. Okay. Then what happens is during REM sleep, areas of the frontal cortex are quiet, but we've got to wake up the hippocampus because, as you probably know from your own experience, your dreams are often your memories. They're set designs. I was in the theater for a while, and I always think of dreams as being they're designed based on your memories. And so you're going to remember your childhood classrooms, or you're going to remember what you were wearing last week or yesterday. But essentially, the hippocampus gives you your memories, and the amygdala gives you that emotional content. And both of these structures are very active during the dream state or REM sleep. And we're going to come back to that when we talk about theories of dreaming, because there's this idea that what dreaming might be is essentially you running back through what you've learned over the last day or two, and trying to integrate it into your other memory systems so that when you wake up, you're going to be more able to survive the world because you've worked out the problems that you've laid down new memories, or you've worked through some of the emotion. And so the thought here is dreaming keeps you in one place because your muscles paralysis, while you reenact the stuff that you had to try to have your brain process. Okay, REM on cells, they're also in the brainstem, in the pons. They use acetylcholine as their neurotransmitter. They're going to be activated when for REM sleep to happen, and then those REM off cells have to go quiescent or quiet. So once again, REM sleep requires that some of the brain increase their activity, but not all areas. So we're going to have those REM on neurons in the pons start having bursts of activity. Okay. What are those bursts of activity? Well, the bursts of activity, first of all, they're going to activate the hippocampus, they're going to start eye movements, and they're going to trigger something called PGO waves. PGO waves start in the pons, where our REM on cells are. They're going to go to the geniculate. Don't know if you remember, geniculate was a subpart of the thalamus. So that's where we're going to pull in some visual information and some sensation as well. So we've got, remember that thalamus, really important for integrating upper cortical sensory information and lower brainstem information. And occipital, That's where vision is. That's where vision is stored. That's where visual memories are. And so these PGO waves travel from the pons through the thalamus to the occipital cortex. And essentially, these REM-on neural clusters are in the pons, in particular reticular formation areas in the pons. So that reticular formation REM off has to be inhibited for REM sleep to happen. Okay, so reticular formation, we're going to inhibit it so we can go to sleep, and then a small portion of it has to be activated for REM. So I hope that's not too complicated. So we've got small areas of REM off cells, mostly in the raphe nucleus. We've got our REM on cells, mostly in the reticular formation, but all the other area of the reticular formation has to remain quiescent or quiet for you to stay asleep. So how do we know about these PGO waves? Because we can actually study them, and we've studied them in cats because all mammals show the same kind of dream patterns we have. And so this is a recording from the lateral geniculate nucleus, LGN, of the thalamus. And here's a cat's brain stem right here, this green area. It's reticular formation, sits in the pons. If you notice, it doesn't exist at quite the same right angle that our brains do, and that's because a cat walks on all fours. And so their brain sits a little differently than our brain, but the structures are all the same. A cat has an occipital cortex where vision is processed, has a diencephalon with a thalamus and a hypothalamus, has a pons where our reticular formation is. And then you can see what happens in a REM state. We get these PGO waves, or a change in the EEG that allows us to basically understand that we're in a different stage of sleep. Okay, so we had REM off cells and REM on cells, and now I'm going to add one last complication. REM on cells can be tonic or they can be phasic. And so tonic refers to steady activity. So there are some REM on cells that are active and stay active all during REM sleep, no other time. Then there are other types of cells that show bursts of activity, and they're responsible for these phasic things like PGO waves. So two different types of REM on cells that look different activity-wise, but they're both active during REM sleep. They're not active during non-REM sleep. And REM sleep's really difficult to coordinate, really, because we have all these neurotransmitters that we have. Glycine has to go up for that muscle paralysis. We have to keep the rest of the brain quiet, GABA, but we have to wake up certain levels like the hippocampus and the amygdala. And so that's really complicated. And if you remember, we also have that serotonin activity in the raphe nucleus. Well, that's important for non-REM sleep, but then we actually have to shut that down for REM sleep to come in. So we've got changes that have to happen, and we've got to once again coordinate that switch. We got to have that flip-flop circuit, not just for waking and sleep, but for non-REM and REM. Okay, and so what we often think about is reciprocal inhibition. And so the idea here is that REM on areas may gradually stimulate REM off neurons that then turn REM sleep off, so that we can cycle back through non-REM. In other words, it's a circular pattern, and that's probably what's explaining why we have components of non-REM and REM sleep every 90 to 100 minutes, and we have cycles, so we're alternating back and forth. And as these neurons kind of like a light phase in and phase out, phase in and phase out. But there's some things that we don't understand why they happen, because science, we don't know everything yet. So we don't know why the REM periods get longer as a proportion of that 90 to 100-minute sleep cycle as your sleep period goes longer. So in the early morning hours, more proportion is REM, and we don't know why. REM also, those REM on cells, REM off too, but REM on primarily, well, it's all in the brainstem. So does that mean REM sleep was actually the thing that we evolved first? Because the brainstem's more rudimentary, doesn't have the ability to executive function like the basal forebrain or forebrain does. So if the basal forebrain starts non-REM sleep, but REM sleep starts in the brain stem, the brain stem's more rudimentary. That suggests REM sleep came first. And we don't get that. We don't understand why that would be. And then, of course, we do know that many sleep disorders basically occur because of some kind of imbalance in these shifts. It's not happening correctly. So sleepwalking, sleep talking, narcolepsy, REM behavior disorder, all of these different things. It takes a lot of coordination to dream and flip back and forth between non-REM and REM sleep. And that's where we have our sleep disorders. We don't know exactly what's going wrong, but and it also makes sense that sleep disorders tend to happen more in young children and old adults, basically as the brain's trying to learn this reciprocal inhibition flip-flop circuit, and perhaps as it starts to wear out as we get older. Okay, so we got the hypothalamus, we got the pons, we had that basal forebrain, but remember I kept talking in the hypothalamus about the SCN, suprachiasmatic nucleus. Well, it sends information to the pineal gland, and that of course is where melatonin is released. And here is where basically that hypothalamus, this upper midbrain, this upper midbrain area is where we can link circadian and sleep processes. So the SCN, it's our master clock. It basically responds to daylight cues through neurons communicating that information from the eye. It helps coordinate the release of melatonin, and that melatonin then can diffuse throughout the body and help basically signal these internal master clock times. And what's interesting is it's not just a diurnal cycle. Melatonin levels and SCN activity also change seasonally. So we have seasonal cycles as well. So once again, it's a master clock, it's a timekeeper. It helps control protein synthesis. So when we're going to make certain proteins like growth hormone, we'll talk about that later. Now we can start to shift that clock when we fly somewhere, when we have jet lag. That's basically your body saying, my internal clock doesn't match the outside world, and that's a problem. It slows us down. It makes our body not as resilient. Blind individuals actually have a lot of difficulty establishing strong circadian rhythms. They tend to report a lot of insomnia and sleep fragmentation, and it may be because this SCN isn't getting the required signals that they need. So once again, the SCN in the hypothalamus, so it makes total sense because the hypothalamus controls hormone release. Then we've got this control of circadian rhythms. And it's all about coordination. So we have little structures that are definitely processing specific things. The hippocampus, the amygdala, each is processing a specific thing, but remember that we have to relay those messages. And some of that is relayed via neurochemistry, melatonin release, for example, and some through neurotransmitters, but we have to project. We can't just call up another brain area. We actually have to physically project neurons to travel and to exist in space so that the message can travel along that neuron. And just as one last side note, just to be clear, your brain isn't all of one thing. So not all brain areas are equally active during the day. That doesn't mean you don't use all your brain. That's a misnomer to say you only use like 10% of your brain. But some brain areas are more active than others, and those are areas that might need more sleep. And so have you ever just been kind of daydreaming or phasing out or not thinking fully? Maybe you're driving and you didn't actually fall asleep. You didn't have a micro sleep, but you don't remember passing that exit 2 exits ago. Well, maybe that's because on some level, one area of your brain was actually having a micro sleep. And so it looks like these different cell assemblies, different areas in your cortex may have different needs based on how active they are. And so these bottom up little structures that did a lot today and are feeling sleepy and need to shut down, they have to coordinate or basically influence your behavior just as those top-down processes, your basal forebrain area and the zeitgeibers that coordinate sleep on a whole level. And so we have to integrate these two things, but it can also be why you have some days where you're really alert because your brain is kind of a little bit better at having gotten rest versus some days you're not so alert, and that's because maybe those brain areas that really need sleep are essentially working as bottom-up influences to lower your overall arousal.